So let's see how eukaryotes can regulate their gene expression. As I mentioned earlier, it is more complicated. It is more complicated because in eukaryotes, for one thing, DNA is enclosed in a double membrane, the nuclear membrane. The DNA is coiled around histones, which are not present in bacteria. And these genes, they are all tangled up with these proteins and are very condensed. All genes are not available for transcription. If we want to transcribe a gene, first we will have to change the structure of chromatin. So we need additional proteins to do that. I would like to introduce you to two types of chromatins, euchromatin and heterochromatin. I have mentioned this before, that the chromatin which is basically used to transcribe is called the euchromatin, and you can see the lighter parts in the chromosome are actually the euchromatic, whereas the dark bands you are looking at are the heterochromatic. In heterochromatic regions, the DNA is very tightly coiled and is not available for transcription. Additionally, I showed you this figure also previously. This is a bar body, an inactive X chromosome, which is highly condensed. The more the DNA is condensed, the darker it stains with this technique. So these darker regions basically are implying that DNA is no longer available in this region for transcription. So let's continue. In order for DNA to become available for transcription, there has to be changes in the chromatin structure. We know chromosomes are made up of proteins and the DNA. DNA is wound around these histone proteins, and these structures fold over and over each other, forming the chromosomes we look at when we see them during cell division. Even without cell division, the chromatin is still very coiled, highly coiled. So the first thing we will need to do is open up DNA, which becomes available to the transcription machinery. In order for that to happen, protein, an activator protein, must interact with the chromosome or the DNA at the right location. This happens when the cell receives a signal, external signal, that now it needs to make a certain type of protein. In our previous example, the signal was presence of lactose or absence of tryptophan. Both of these were sensed by the cell, and in response to these two changes, cell responded by altering the transcription of its genes. Here, in eukaryotes, when a cell receives a signal, it activates a transcription activator, or it produces a transcription activator. It binds and it recognizes specific regions of DNA, and it binds with it. This is the starting point from where on the transcription will take place. One of the things that have to happen is removal of the histones. Chromatin remodeling complex is recruited by the activator. Chromatin remodeling complex, or CRC, one of the things it does is it puts the acetyl groups on to the histones. As we know, acetyl groups are negatively charged. When these groups are placed, here you can see the blue dots on this figure are basically the acetyl groups. These acetyl groups, because of their negative charge, repel the DNA, which is also negatively charged. So DNA loosens up, and now it is available to interact with the machinery that is responsible for transcription. But the things don't end here. How eukaryotes, requirement for transcription of eukaryotes is still different. We saw in prokaryotes, RNA polymerase could directly bind the promoter. That is not the case in eukaryotes. We will see how RNA polymerase is able to bind the DNA with the help of other factors.